it is a pleasure for me to introduce the next speaker. It is a Professor Ralf Metzler, Universität Potsdam, and the topic is fractional transport processes, search, tempering, and data analysis. Please.
Okay, thank you very much. Thank you for the invitation. Only from last week, my university would have allowed me to travel to UK again. So this is why I can't be present in person, because there was no way to plan this trip. My internet connection is pretty bad, so I hope it will survive this talk. Before I start, I want to mention briefly that actually for her PhD work that Vittoria presented, she got the Mate Vogt Award for the best PhD thesis in Berlin. So I think that's worth mentioning. The second thing, I would like to follow Enrico in dedicating this talk to Mark Metzler, who was a remarkable mathematician interested in applications, actually, with all his work in kind of geophysics and other systems. And he had a very long and interesting, in a good sense, battle with cancer. And if you're interested, there's a beautiful article on the American Cancer Foundation. If you Google Mark Metzler battle and cancer, you will get that easily. Okay, let me come to my talk. I changed the title and the content a little because of a result we recently got, and I want to talk about different approaches for fractional transport in confining potentials. And before I get there, I want to go back into history. Old men like to come up with historical sources. So I want to feature this article by Eugen Kapler, one of the pioneers of diffusion measurements. And you know the Einstein relation, the connection between the diffusion coefficient and thermal energy. And with his experiments, Kapler actually managed to extract Avogadro's number from his experiment to a remarkable accuracy. He used a rotating setup. So there was a small mirror that could rotate, but the mirror was suspended on a quartz thread, a very thin quartz thread. And this is why when you look at the trajectory that is created by the reflection of a light beam on this rotating mirror, you can see that it doesn't look like free Brownian motion, because it's not. It's experiencing a restoring force due to the suspending quartz thread. And the angles are pretty small, so we can talk about a harmonic approximation of this restoring potential. And what he did as part of his experiments, he also just measured the equilibrium distribution caused by this rotational Brownian motion. And he actually managed to map out this beautiful Gaussian. This is, of course, the Boltzmann distribution. And in a harmonic potential, it is a Gaussian. So we'll come back to confinement of fractional processes in harmonic potentials, but especially I want to look at non-harmonic potentials. I want to look at potentials of this shape. So they're all confining, and we will say, I mean, they have a confining tendency, let's say. I will show to you that not in all cases these potentials actually manage to confine the process. And I especially will distinguish between potentials that are steeper than a harmonic potential and those that are shallower. Let me quickly say a few words. Today's harmonic oscillators, of course, in physical systems, they are the most prototypical example when we think of anything connected to a potential. And we can have this, as I just said, the harmonic potential as a first order in a symmetric potential when we neglect higher order terms of order x to the 4 here. I have no clue where this s comes from, please ignore it. A Hookean force can be realized experimentally by optical tweezers. or by anchoring a tracer particle to a flexible polymer chain. These are experiments that were performed in many labs. Steeper potentials, they might come as higher order terms in the confinement potential when this becomes relevant, or as ever better approximations of a steep box-like potential. And shallower potentials, on the other hand, they can come from entropic forcing in confining channels. You can have symmetric linear potentials in many textbooks as prototype examples for non-harmonic forcing. They're realizable in holographic optical systems, and the extreme case, a logarithmic potential, this is what the next speaker has been working on extensively. They may be connected to laser systems. Finally, potentials of different forms can be used as models, in a more abstract sense for home ranging of animals in a habitat. They might represent degrees of lockdowns and all these games that we're playing. Okay, let me start with maybe a partial answer to the question we just had. What if the kind of diffusing diffusivity does not come directly from the environment, but it might come from the particle itself? So I show you one example here. This is a relatively simple protein, and you see here results from extensive computer simulations. This is work with Eiji Yamamoto and Takuma Akimoto. Eiji Yamamoto, did I say it correctly? Okay. And you see that this protein can assume very, very different shapes. And these shapes correspond to local minima of the potential landscape. So these are generally not harmonic, but maybe one can approximate them as locally harmonic little potential wells. And the protein is able to move in this landscape and then have more extended configurations, more compact configurations. And as you can see in this in this time series down here, we have the time local, the instantaneous size of the protein. So it's fluctuating. Here we have these very extended configurations. Here we have the more compact ones, and you see it's fluctuating. And when the hydrodynamic radius is large, of course, also the mobility will be decreased. And you see here in the same plot, the gray lines to the bottom, this is the time local diffusivity. So we have here an example where the shifting shape of the tracer itself is causing these fluctuations, this randomness in the diffusivity. If we go to more complicated proteins, now this is a work from Sunny Shi's group many years ago. They looked experimentally at, as I said, more complicated proteins, and they had 2 amino acids that they tagged, and they could then measure the relative motion of these 2 amino acids. And they actually managed to map out that it corresponds to something like the relative motion is happening in a harmonic confinement, because of course this is a finite chain, it's a polymer chain more or less. So when they fluctuate, it needs rearrangement of the whole chain, and it can't move too far away because there's a finite extension that's possible. However, the dynamics is no longer exponential. It has anomalous diffusion, and they actually fitted it with the fractional diffusion equation in that first paper. There was a long subsequent discussion, but it's actually getting back to this interpretation like a CTRW, like the particle motion gets trapped before it can continue to move. And the naive idea people had in that paper was that it's not a pure harmonic potential, but you have a zigzagging on top of the potential that causes these local arrests. The same system was simulated a few years ago in the group of Jeremy Smith at Oak Ridge, and they, of course, had much more extended data available. They had the whole time series of all the positions. And the idea, again, here in my bad cartoon, this is my protein chain, and I'm looking at the relative motion of 2 amino acids, of 2 monomers in this chain. And what they see is the time average mean square displacement has a very peculiar shape. It especially does not reach saturation, something you would expect from an ensemble average, and I will come back to that in a second. They also see that the correlation function of the motion has a shape that is dependent on how much of the trajectory you analyze. The measurement time dependence of the characteristic time, for instance, to decay to 1/e is plotted down here, and you see in a log-log plot, the characteristic time scales with the measurement time. And they also have some statements in their paper that they observe power law waiting times in the motion, in the relative motion of this protein, because there are arrested configurations. Now, one way to describe this is by these time-fractional Fokker-Planck equations, where we have here the confining potential, here we have the diffusivity, and this is a Riemann-Liouville operator. And what we showed together with Eli and Stas Burov quite some years ago was what happens to the time average in this confinement. First of all, it's universal. The result only depends on the first two moments of the Boltzmann distribution of the corresponding potential. And then at very short times, you have in this weakly non-ergodic system, a linear slope of the time average mean square displacement. But at intermediate times, once the motion engages with the confinement of the potential, you see that we have a lag time dependence to the 1 minus alpha, whereas the ensemble average would actually show saturation. So that's looks in a way a bit similar to what they see here, but of course the motion in the simulations is more complex and also most likely truncated. There's also some aspects of aging that we were discussing together with Eli and my PhD student Johannes some years ago in these papers. And experimentally, very likely this is what was seen in an experiment with optical tweezers focusing on lipid granules in living yeast cells. And you see indeed here this linear slope of the time average mean square displacement at short times, and it crosses over to this shallow slope that corresponds to 1 minus alpha. So this is what happens when we look at this classical subdiffusion that can be described by these time-fractional equations. Now I want to go to the exact opposite and show some older work, and then I will connect this to more recent work. Now I want to go to the opposite case when the dynamic is perfectly Markovian, but we have these space fractional diffusion effects, and they are of course related to power law distributions of the jump lengths, something that is typically connected with either motion, effective motion of units on fast folding polymer chains, or on a more macroscopic scale with the motion of animals and whatnot. And again, I want to consider these potentials. When the potential is harmonic, you get a stationary state, but the stationary state has the same Levy stable index as the driving process. This is connected, of course, to Levy stable noise driving the motion. And this we showed many, many years ago. What happens when we go to a steeper than harmonic potential? Let's say we have a quartic potential. This is work we did with Alexei Chechkin, Vsevolod Gonchar, and the old grandmaster of Kharkov, Leonid Tanatarov. And what we saw was actually something very surprising. When we vary the mu, the Levy index, we can actually create multimodal states. So when mu is 2, this is the Gaussian process, this is just the Boltzmann distribution for a quartic potential. And then once we lower the Levy index and we emphasize these more extreme jumps, then we go towards this very clear multimodal distribution. Here you can also see it as a function of time. We start with a more concentrated initial condition. The thin line is the Boltzmann distribution, and then you see how we have these emergence of the two peaks. And here, as a function of time, you can see this phase diagram. We have a single peak, and then it splits up into two peaks. There's also more complicated situations when we have intermittent situations of 3 peaks and so on, which is discussed in these older papers. We can also use a different potential, this arcus-tangent potential that looks like this, I think, orange line here. So it approximates a box potential, and you can see very, very complicated crossover behaviors with multimodal states here. Now, what happens when we use the same potential, the same equation, but now the C becomes smaller than 2? So we have shallower potentials than harmonic. And there in some work, where's my mouse here? In some work some years ago by Dibiec, Alexei Chechkin, and Igor Sokolov, they showed that stationary states only exist as long as this inequality is fulfilled. So the potential needs to be sufficiently steep to confine the Levy flight, because it has this tendency to venture very far out, and the potential has less and less power to actually confine the motion when you lower the C. When mu is 2, we have a Gaussian process. You see that any positive C will produce a stationary state. And once you lower the value of mu, you can see that you have finite values here that you need to overcome by the C. Now, let me switch to another fractional process, and this is fractional Brownian motion, and you will see that there's many, many similarities in how the system behaves. So now we describe the process with this Langevin equation. Here we have our confining potential, and here we have a fractional Gaussian noise. So it's Gaussian, but it is long-range correlated. And I show the correlation function. Here you see that when we have a subdiffusive process, when alpha is smaller than 1, these are negatively correlated. When alpha is larger than 1 and we have a persistent, a positive correlation, then of course this value is positive. Let me show you the result for a quartic potential first. When we have a superdiffusive process, alpha equals 1.8 in this case, you see that again, we start with a localized initial condition and it ventures out. It still looks more or less Gaussian, but all of a sudden, when it engages with the wings of the potential, it creates this bimodality, similar to what we saw in the Levy case. It's less clear what happens here for the subdiffusive case, but if you look carefully here, for instance, for this yellow line, the thin line is a Gaussian approximation, and the symbols of the process, they are narrower a little. So here actually the probability density becomes a bit more concentrated than the Boltzmann distribution. It's a bit more clear when you look at a box potential with an infinite slope. You see here the time evolution for Brownian motion. Eventually it has the equidistribution that we would expect. For subdiffusion, you can very clearly see here now how it's actually depleting at the boundaries. And when you look at the sample trajectory here, you can see that this anti-persistence, when it hits the boundary, it's reflected back, and then the next step, due to the anti-persistence, still leads away from the boundary. You can clearly see it doesn't want to touch the boundary. This is the case for a Brownian motion, and this is the case when the motion is persistent, when it's trying to push against the boundaries. And here, in this case, you can see this very clear increase of probability next to the boundary. So we have, again, multimodality here, and here it's due to this, due to the presence of the positive correlation. Now, in a paper that we're going to submit in a few days, we looked at the question again, what is if we have a shallower than harmonic potential? And here we evaluated the whole process in terms of the probability density function, but also in terms of the mean square displacement. And you can see here, for instance, in this plot, when we vary the alpha, you can see for smaller alpha that we have a convergence, the mean square displacement reaches a plateau. But once we go to large enough alpha, the mean square displacement just continues to grow. Again, we reach a case where there is no confinement, where there's no stationarity of the system. And we get, we conjecture from the analysis that we have such an inequality where alpha is the anomalous diffusion exponent. But if we introduce a self-similarity index of the motion, we introduce this Hurst exponent, this corresponds to alpha over 2, then we can rewrite the condition as this. And then if we use the same self-similarity exponent for a Levy flight, we see that actually this inequality has exactly the same structure as for Levy flights, which is quite a remarkable property that I like very much. Now, can this be relevant for experiments or for real systems? And this is, here's some examples from a work with Schemantos Janushonis, who is a biologist at Santa Barbara, and also with Thomas Feuta, a theoretical physicist from Missouri Roller. And so we're interested in what happens in mouse brains, and this, of course, should translate also to other mammalian brains. And Schemantas is looking at the spatial distribution in these cross-sections of so-called serotonergic fibers. These are axons. They're very thin axons, and they exist in all mammals. And what they see is once they smoothen everything, that the concentration of these fibers is very high, close to hard boundaries of the brain. So this is a very typical property, and for some reasons, we assume that we can model the growth of these fibers by fractional Brownian motion. Here I show you an example between traces of these fibers in these cross sections, and this is a simulation of a fractional Brownian motion, and you can see, well, at least there's a qualitative similarity. Now, once we go and simulate these kinds of shapes, you see for subdiffusion what I told you, we see a depletion. There's less concentration around the boundaries. For normal diffusion, everything is nicely, evenly distributed. And once we go to the super diffusive case, we see exactly corresponding to the experiment, we see this enhancement, this increase of concentration next to the boundary. I don't see a clock here. How am I doing in time?
Let's say you're a bit less.
I'm a bit less.
5 minutes, yeah.
It
depends how much time do you want for questions, but for sure 5 minutes is fine.
Perfect, thanks. So the final few words I want to just mention, I will not be able to talk about this in detail, but we also see multimodal states in Levy walks. Levy walks, as many of you know, are spatio-temporally coupled processes. So, for instance, you can have still jump length distribution that is long-tailed, but every long jump is punished by a long time consumption. It has a finite speed, and still we can see multimodality in these processes, and they even occur in harmonic potentials in this case. And also we recently looked at what happens when we reset a Levy walker. Here, resetting is not the direct move of the walker at a given random time back to the origin, but what we do is at a given time, a random time, we switch on a harmonic force, and this is slowly pushing the particle towards the origin. And in several cases, when we vary the parameter space, we again see multimodal distributions. The fact that fractional Brownian motion, persistent fractional Brownian motion, is actually related to the movement of animals, we actually analyzed in a recent paper with many co-authors, many experimentalists, colleagues. This is actually this is being reviewed as we speak. And for instance, for the flight of certain birds, here you can see the trajectory of a stork very close to my office next to Berlin. We looked at different scaling exponents in the trajectories, and we actually could not confirm for these cases that the motion is related to Levy walks, but it was actually much more consistent with persistent motion with fractional Brownian motion with a positive persistence. I mean, here you also see some example of a vulture moving in Israel and then down along the Red Sea. And we have a lot of information about these processes, and we typically do not see Levy type statistics. Very short commercial, we're also looking at data analysis in our group. especially also for anomalous diffusion, for fractional motion. We use Bayesian analysis, and if you're interested, I'm very happy to answer questions. And very recently, we organized, the main organizers were Giovanni Volpe and Carlo Manzo. We organized the anomalous diffusion challenge. Here you have data, tell us what kind of process is behind the data. And soon there will be a second round of this. So if you're interested in competing with other groups on data analysis, watch out for anomalous diffusion challenge number 2. So what I showed you, Levy flights and superdiffusive FBM, they have non-Boltzmann statistics. They have multimodal PDFs when we have superharmonic, steeper than harmonic confinement. When we go to the opposite case, when we have shallower than harmonic potentials, we actually have a critical exponent for the potential. And if the potential gets too shallow, it can no longer confine persistent fractional Brownian motion or Levy flights. So that's quite a remarkable property, and especially both processes share the same value for the critical exponent if we express it as in terms of the similarity index. Fractional Brownian motion next to hard walls or steep potentials, they show this accretion or depletion, and it could be relevant for systems as the one I showed you before. Also, Levy walks have multimodal PDFs and even in harmonic potentials. And what I didn't have time to talk about, we also have some recent work on first passage and first hitting statistics for different processes, especially also for Levy flights and Levy walks. I want to acknowledge at the end all the funding sources over the last couple of years and all the friends and colleagues with who I enjoyed collaboration. And let me say at the very end, I just want to meet everyone in person again. Thank you.
Thank you very much. Brilliant summary. Thank you. Do you have any questions? Yes, please.
Hello, Ralf. Thank you for your talk. I'm interested in filling in a little bit more of the detail about the link between the data and some of the models that you've been using. Towards the end, you summarized some things which I think get close to that, where you were talking about the Bayesian approaches and so on. But in some of the earlier work, we went quite quickly from diagrams of trajectories of data points across to your model equations. And I was just wondering what your approach has been, whether it's very much the matter of matching the qualitative behavior or whether you've had a more systematic approach.
Well, the very systematic approach at the beginning was actually, we were scribbling down equations and we saw these strange behaviors. This is what it all started like. But of course, over the time, we also got a bit more serious, and especially when I'm not talking about Levy flights anymore, but when we talk about Levy walks. But the most closely related to experiments is fractional Brownian motion. So is a very typical behavior we can see in biological systems. When you go to subdiffusive FBM, that's typically the kind of statistic you see for traces in crowded systems. So there are quite a number of experiments that show this kind of behavior. And then you can combine it with confinement, for instance, by creating obstacles, by creating an optical trap around them. And for the superdiffusive FBM, it's becoming more and more a model for when particles move, for instance, in cells. So you have little motors, whatever. And what I showed you, it also seems to be relevant for large animals. And there the confinement could be, you know, here I have to fantasize a little bit because we don't have enough data and we haven't done the analysis properly. But of course, the animals eventually go back to their habitats, they go back to their nest or whatever. The parents call back the offspring when they wander about. So this could be kind of a more heuristic argument.
Thank you very much. Okay, we have one more question online, and that's.
I guess it's me. Huh? Hello, Ralf. Nice to see you again. Just one remark. I think in a way you said that, but I suppose one can interpret the multimodality of the distributions more generally as a stickiness phenomenon of, say, persistent motion to the walls, which is also very well known for, say, active Brownian particles, though these active particles are not typically modeled by FBM, right? But this is a general context, right? One knows that the particles are persistent. They start to accumulate at the walls, and if you have smooth walls, okay, then it's not too unreasonable that you see that as these peaks in your distribution, right? This is probably the general context, and related to that, I mean, probably it's some of your papers, but is there an analytical solution of this problem, really? I mean, I think you have shown simulation results, right? But can you really solve these equations with respective boundary conditions or potentials?
I mean, fractional, sorry, I interrupt you, Rainer.
No, I'm
fractional Brownian motion, you know, is a terrible process to approach analytically. So no, we don't have exact solutions here. It's really simulations based, but at least, you know, what these results show is why we cannot easily calculate first passage problems for fractional Brownian motion, because it doesn't have a horizontal derivative next to a reflecting boundary. All these correlations, you know, they make the process so nasty. Of course, you're right that many active processes have the same kind of features, that you have this, what you call stickiness or accumulation. But I was still, you know, quite surprised, for instance, about this criticality we have now and this similarity to the Levy motion. So there's quite some stuff that still can be explored. And, you know, maybe if we use these Gaussian processes to at least model some features of active motion, maybe, you know, there can also be some exchange with this kind of physical models.
Yeah, in biology they call it correlate random walk, which goes in this direction. So that's what they've been using, but also you know this, I guess. Okay, thanks a lot.
Yeah.
Okay, thank you very much. Let's thank speaker again.
Thank you.
And it is my pleasure to introduce the next speaker. It is Prof.